Hello, Miss Mayhew here, and we're going to continue talking about DNA and its role in heredity. Today we're going to focus on the way that DNA replicates, which is semi-conservatively. Okay, semi-conservative replication means that each of the parental strands is a template for a new strand. Now we can simplify DNA replication into two general steps. The first one is that the double helix is unwound, making those template strands available for new base pairing. Then, free nucleotides form base pairs with the template strand and are linked together by phosphodiester bonds. This diagram shows the addition of these nucleotides to the growing strand of DNA. So in this first diagram, you can see that there's a cytosine that is going to be attached to a guanine where it matches, and we are forming a phosphodiester bond between the 3' end of the strand and the 5' end of the newest nucleotide. So during DNA synthesis, because we have to form those phosphodiester bonds between the nucleotide bases on the new strand, these new nucleotides are only added to the 3' end of the new strand because there is a free hydroxyl group so that we can perform a dehydration synthesis reaction in order to form that phosphodiester bond. The DNA template is read from the 3' to 5' end, and the new strand is generated from 5' to 3', forming this anti-parallel double helix. Now, as we're building the DNA strand, it's synthesized from these deoxyribonucleoside triphosphates, or deoxyribonucleotides. These are basically the nucleotides with 3 phosphate groups. So during synthesis, 2 of those phosphate groups are released, and the final nucleotide is a monophosphate. So these are the adenine, thymine, cytosine, and guanine nucleotides that you're used to seeing, with the 1 phosphate group, the 5-carbon sugar, and the nitrogenous base. When we release those 2 outer phosphate groups, that's what provides the energy to form the phosphodiester bond. DNA replication begins with the binding of a large protein complex, or a bunch of proteins that work together, that we call the pre-replication complex. That binds to a specific site of the DNA molecule that we call the origin of replication. This protein complex contains DNA polymerase, which catalyzes the addition of nucleotides. In prokaryotes, the pre-replication complex binds to the origin of replication, and the DNA unwinds, and replication proceeds in 2 directions. The replication fork is the site where the DNA unwinds. Opening each fork is catalyzed by DNA helicase. So in this diagram, at the top, we see a prokaryotic chromosome. Okay, it's circular, and we have the origin of replication highlighted in purple. So the first thing that happens is that pre-replication complex is going to bind at that origin of replication, and the DNA is going to be split apart. You can see that it's split apart in 2 directions. It's opening up both ways, and those are called the replication forks. Now, the enzyme that does this on both sides is called helicase. DNA replication in eukaryotic cells, on the other hand, they contain longer linear chromosomes. So they have multiple origins of replication. This allows the opening and replication of DNA to occur a little faster. So like I said, eukaryotic chromosomes are longer and linear, so they need multiple origins of replication. That's going to speed up that process. Okay, if we only had 1 origin of replication, it would take weeks to fully replicate that chromosome. Now, in DNA replication, as we start to build the new strand of DNA, this begins with a short primer or a starter strand. Usually, this is a short single strand of RNA. This primer is complementary to the DNA template and is synthesized by an enzyme called primase. This DNA polymerase then can add nucleotides to the 3' end of the primer, because in order to add a new nucleotide and form that phosphodiester bond, we need that free hydroxyl group at the 3' end of another nucleotide. So that's what the primer does. Then it's going to continue doing this until the replication of that section is complete. So here you see that my primase has added a green primer. Then the primase is going to detach from the DNA, allowing DNA polymerase to bind to the DNA. Now it can add new nucleotides, starting with that 3' end of the new strand and moving along the DNA molecule. Now, DNA polymerase is a very large, complex enzyme that's shaped kind of like an open right hand. The palm part of your hand would bring the active site and the substrates into contact. The fingers are going to recognize the nucleotide bases, and the enzyme is then going to change shape and catalyze formation of a new phosphodiester bond. So here's a diagram of what they mean by that. Okay, you have the thumb, you have the fingers, and you have the palm where the DNA is kind of laying. That's going to allow that DNA polymerase to change shape and catalyze those phosphodiester bonds. Now, although there are 2 replication forks, each replication fork only opens up in one direction. The 2 new DNA strands are anti-parallel, so the 3' end of one strand is paired with the 5' end of another. But DNA replication occurs in a 5' to 3' direction only. That means that each of these 2 anti-parallel strands are going to be replicated differently. Okay, one strand is going to be called the leading strand. It's oriented to grow continuously at its 3' end as the fork opens up. So that means it's opening up in the same direction that DNA polymerase is moving and adding nucleotide bases. The lagging strand, on the other hand, is anti-parallel to that, so it's oriented so that the exposed 3' end moves further away from the fork. Synthesis of the lagging strand happens in small discontinuous stretches called Okazaki fragments. Now, looking at my top diagram of DNA replication, I can see that my primer has been added and this new strand of DNA is moving in the 5' to 3' direction. That means that as my replication fork opens up, there's always a new 3' end for DNA polymerase to add a new nucleotide to. This strand is going to be my leading strand because it can be done continuously. Now, looking at my second diagram, I see that I have a 5' end that's available. Unfortunately, DNA polymerase can't add new nucleotides to the 5' end. So what we have to do is we have to add a primer as it opens up. This allows my DNA polymerase now to attach new nucleotides at the 3' end of the primer, moving in the opposite direction of the replication fork. Now, looking at my third diagram, as my DNA polymerase is moving along and forming these Okazaki fragments, my replication fork still continues to open. So as it opens more, that gives us room to add yet another primer and continue this until the DNA is completely formed. Each of these Okazaki fragments is going to need its own primer so that each time DNA polymerase starts, it has a 3' end to add to. DNA polymerase is going to add nucleotides to that 3' end until it reaches the primer of the previous fragment. Then a different DNA polymerase is going to replace the primer with DNA, and the final phosphodiester linkage between those fragments is catalyzed by DNA ligase. So here at the top, we have our primer being added. Next, we have those nucleotide bases being added by DNA polymerase. Then a different DNA polymerase is going to replace that RNA primer with DNA nucleotides. And then the ligase is going to form that final phosphodiester bond between the first and the last nucleotides of each of the Okazaki fragments. DNA polymerase works very fast, but makes very few errors. It's processive, which means it catalyzes many sequential polymerization reactions each time it binds to DNA. A DNA polymerase can add thousands of nucleotides before it detaches from DNA. DNA polymerase can make mistakes in the replication process, but most of these errors are repaired. There are two major repair mechanisms that DNA polymerase uses. The first one is proofreading. DNA polymerase has a proofreading function, and if bases are paired incorrectly, the nucleotide is then removed. Mismatch repair is another repair mechanism that after replication, other proteins scan for mismatched bases that are missed in proofreading and replace them with correct ones. So not only can DNA polymerase check itself as it goes and remove the mismatched nucleotides, but there's also a second DNA polymerase that is going to do mismatch repair. Now there is a little catch. When that last primer is removed, no DNA synthesis can occur because there's no 3' end to add to. So there's a single-stranded bit of DNA that's left at each end. Those are cut and the chromosome is slightly shortened after each replication. Now these chromosome ends have to be protected from being joined to other chromosomes by the DNA repair system. Each eukaryotic chromosome has telomeres, which are repetitive sequences at the ends of those chromosomes. These repeats bind with a protein complex called shorten, which protects the ends from being joined together. The repeats also form loops, which are protective as well. So in the top diagram, you can see what we talked about like 2 slides ago, where in most cells, that terminal non-replicated part, after we've removed the primase, is just going to be removed. Okay, that shortens the chromosome. But in certain types of cells, like stem cells and cancer cells, there's an enzyme called telomerase. This uses an RNA template to extend the telomere. Telomerase moves the new end and DNA polymerase fills in the gaps. This process can be repeated multiple times to lengthen the telomere. Now, in our regular cells, after about 20 to 30 cell divisions, the chromosome ends become too short, and the chromosomes lose their integrity, so apoptosis ensues. This is where the cell is purposely destroyed. But continually dividing cells, like bone marrow and gametes, maintain their telomeric DNA. This is because telomerase catalyzes the addition of lost telomeric sequences, and it has an RNA sequence that acts as a template for that telomeric DNA. Because each cell division shortens the length of a chromosome, the telomere length tend to shorten as you age. So if a gene expressing high levels of telomerase is added to human cells in culture, their telomeres do not shorten and the cells become immortal. This is also seen in mice that overexpress telomerase. They live longer. Cancer cells also express telomerase. Some really cool gene technology that we're going to talk about real quick is polymerase chain reaction, or PCR. This is where copies of DNA sequences can be made from a double-stranded DNA sample. You can take 2 primers complementary to the ends of the sequence that you're going to try and copy. Combine that with the 4-deoxyribonucleotide triphosphates, a DNA polymerase that's going to work at high temperatures, and salts in a buffer to maintain the pH. PCR is a cyclical process in which a sequence of steps is repeated over and over. DNA replication is fast, so it takes a really short time to make millions of copies. The sequences at each end of the amplified fragment must be known ahead of time so that the complementary primers can be made. Once you have a pair of primers, that will bind to a single region of DNA in the organism's genome. So what this looks like is we have our target sequence, and we have the primers that match each end. We heat this up in order to break the hydrogen bonds and split our DNA. The primers are going to attach. Our DNA polymerase that works at the high temperature is going to head to go ahead and fill in the rest and make 2 new strands. Now, every time we do this step, we're going to double the amount of DNA that's been made.